Morning, dear student. In today's lecture, we will discuss the second type of protein ligand interaction, that is allosteric protein ligand interaction. In previous lecture, we will discuss the reversible binding of protein and ligand, that is the reversible binding of O2 oxygen with hemoglobin. So we are very much familiar with this hemoglobin and myoglobin. Myoglobin, again, it is a monomer, while hemoglobin, it is your tetramer. So you should remember this point. The monomer, that is myoglobin, contain only one polypeptide chain, while the hemoglobin containing the four polypeptide chain. Again, we know that this hemoglobin, it is a combination of heme and the globular part of the protein. So heme group contain the porphyrin ring, in which the metal atom is Fe. It is present in +2 oxidation state. Without oxygen, whatever the form of this protein, it is considered as a deoxyhemoglobin, and when oxygen is bound, then it is considered as a oxyhemoglobin. So we will discuss, we in previous lecture we already discussed the differences between this deoxy form and oxy form of hemoglobin. The deoxy form, it is the T state, or we can consider it is as a T tense state, while the oxyform, it is R state, or it is a relaxed state, because there is a lot of interaction such as hydrogen bonding, then ionic interaction, hydrophobic interaction, when when you consider a protein molecule. But whenever a oxygen is bind with this hemoglobin, then these interactions are weak, and therefore which results into the relaxed form of the structure, that is the T state is converted into R state. So we are very much interested in this lecture, that is allosteric interaction between protein and ligand. So what is this allosteric protein? This allosteric protein is one in which the binding of a ligand to one site affect the binding properties of another site on the same protein. So you should remember this point. What is this allosteric protein? It is a protein in which the binding of ligand in which the binding of ligand to one site affects the binding properties affects the binding properties of another site on the same protein, another site of the same protein. So you should remember the definition of this allosteric protein. It is a type of protein in which the binding of ligand to one site affects the binding properties of another site of the same protein. The term allosteric derives from the Greek word, that is, whatever the allosteric, it is derived from the Greek word, that is other. This allosteric stands for others, and stereos it stands for shave. So you should remember, this allosteric means other and stereos means the shape. In other words, we can say that the conformation induced by the binding of ligand is referred as a modulator. The third important point is that whatever the change which is occur after binding of ligands to the protein, the conformation induced by the binding of ligand by the binding of ligand, conformation induced by the binding of ligand is referred as modulator, is referred as modulator. So whatever the conformation which is induced by the binding of ligand is referred as modulator. The conformational changes induced by the modulator interconvert more active and less active forms of the protein. So there is an interconversion between active form to inactive form or inactive form to active form. So the conformational changes which is induced by the modulator means the binding of ligand to the protein interconvert more active and less active form of the protein. The modulator for this allosteric protein, so whatever the modulator or we can say the ligand which is binds to the protein, the modulator for allosteric protein, modulator for allosteric protein maybe either inhibitor or activate. Again, the next important point is that when the normal ligand and modulator are identical, when the normal ligand and modulator are identical, then the interaction is termed as homotropic allosteric interaction. Then it is termed as homotropic interaction. Normal ligands and modulators are identical, then it is considered as homotropic interaction. The best example of this homotropic interaction is the cooperative effect of oxygen, that is the binding of oxygen to the hemoglobin molecule. And when the modulator is a molecule other than the normal ligand, then the interaction is known as heterotrophic interaction. So you should remember, when the molecule other than modulator, then it is considered as, when the molecule or a normal ligand other than a modulator, it is different from the normal ligand, then it is considered as a, whatever the interaction, it is considered as a heterotrophic interaction. Heterotrophic interaction. Some proteins have two or more modulators and therefore can be have both homotropic and heterotrophic interactions. So this is regarding to the allosteric protein. So we will discuss the example, that is homotropic interactions. We are very much interested in this homotropic and heterotrophic interactions. Let us see the first one, that is homotropic interaction. Homotropic interaction. Homotropic interaction. So the best example of this homotropic interaction is that the cooperative effect. So you should remember the example of this homotropic interaction is the cooperative effect. Cooperative effect. So what is this cooperative effect? If you consider the phenomenon of cooperativity in hemoglobin can be explained by considering the corresponding binding binding of oxygen and finding out the rate constant. For example, if we take one, if you consider a hemoglobin, then the hemoglobin is a tetramer. When the one oxygen is bind to the hemoglobin, that is Hb, reversibly bind with oxygen, then there is a formation of oxyhemoglobin HbO2. And let us consider the rate constant for this reaction is K1. Similarly, what happened? The binding of this oxygen, the binding of this oxygen increases the rate of binding of another O2 molecule. The binding of this O2 molecule affects the rate on the second rate constant. The binding of this O2 molecule increases the rate of reaction of second O2 molecule when interact with this hemoglobin. So this HbO2, it will react with O2, then the rate of the reaction is increases as compared to the first reaction. That is, we get HbO2 two times, the rate constant is K2. Again, if you consider when a third molecule of oxygen, it will interact with this hemoglobin, then the rate constant is further increase. So what you get here, HbO2 thrice, that is K3, and HbO2 thrice, it will react with O2 and we get the finally HbO2 4, the rate constant is K4. And if you find due to the positive cooperative among the subunits of hemoglobin, the successive oxygen binding constant increases in the order, that is the K4 is very greater than K3, K3 is greater than K2, and K2 is greater than K1. So why this is possible? This is due to the positive cooperativity among the subunits of the hemoglobin, and this is the best example of the homotropic interaction. So you should remember, whenever uh, the modulator and the normal ligands, so in this case oxygen, it is your ligand and it will binds with your protein, therefore it is considered as a modulator. So the normal ligand is O2 and the modulator are same, then it is considered as a homotropic interaction. And the best example is the uh, cooperative effect of oxygen binding with hemoglobin. Again, we know that similarly, the heterotrophic allosteric interaction involve the cooperative interaction among the different types of substances binding with the target proteins. All these allosteric interactions are absent in myoglobin. So you should remember, whatever the interaction we discussed, that is homotropic and heterotropic, they are absent in myoglobin but present in hemoglobin. So let us discuss the second one, that is the heterotropic allosteric interactions. So at least you should know the definition of this allosteric protein, and then the different types, that is homotropic interactions and heterotropic interaction. In homotropic interaction, you should remember the example of cooperative effect of oxygen binding with hemoglobin, and for heterotrophic allosteric interaction we will discuss the effect of H plus ion, CO2 and Cl minus ion on oxygen binding. So let us discuss the second, that is heterotrophic allosteric interaction. Heterotrophic allosteric interaction. So in this case, the normal ligands and modulators are different, and therefore it is considered as heterotrophic allosteric interaction. The best example is the effect of H plus ions, CO2 and Cl minus ion on oxygen binding with hemoglobin. We know that. So let us discuss the effect of H plus, CO2 and Cl minus on O2 binding. O2 binding with hemoglobin. So both H plus ions and CO2 shows heterotrophic allosteric interaction, due to which increasing concentration of this species reduces the affinity of deoxyhemoglobin towards O2. So you should remember, it is very important, that is both H plus ions and CO2 shows heterotrophic allosteric interaction, due to which increasing concentration of H plus ions and CO2 reduces the affinity of deoxyhemoglobin towards O2, because we know that the deoxy form of hemoglobin interact with O2, and then there is a transport of oxygen from lungs to the various tissue. So you should remember, both if the concentration of H plus ions and CO2 is increases, the affinity of the deoxyhemoglobin towards oxygen is decreases. So you should remember, with increase in concentration, the affinity of deoxyhemoglobin for O2 is decreases. In other words, we can say that the H plus ions concentration is increases, means the pH is acidic, or we can say that the H plus ions concentration is increases, then the pH is lower. So we can write, at lower pH the affinity for oxygen is decreases, or affinity of deoxyhemoglobin towards oxygen is decreases at lower pH. In other words, we can say that the CO2 and lactic acid in the working tissue, the CO2 and lactic acid are produced. Lactic acid is produced by the incomplete oxidation of glucose due to the insufficient supply of oxygen. Thus, in the working tissue, the lower pH stimulate the release of O2 from oxyhemoglobin, and the effect of this pH on O2 affinity of hemoglobin is described as Bohr effect. So we are very much familiar with this Bohr effect, and a very large Bohr effect leading to the sharp decrease of O2 affinity with decreasing pH is known as root effect, which is also the important. Again, the one more example of this heterotrophic allosteric interaction is the effect of 2,3-diphosphoglycerate, that is DPG. The effect of 2,3-diphosphoglycerate 2,3-diphosphoglycerate, it is denoted as capital DPG, on oxygen binding with hemoglobin. So this DPG is it shows heterotrophic allosteric effect, due to which the oxygen affinity of hemoglobin is decreases with increase in the concentration of DPG. So the oxygen affinity is decreases with increase in the concentration of DPG. So you should remember. So whatever the effect of H plus ion CO2 and the effect of 2,3-diphosphoglycerate, that is DPG, it is inversely proportional. So we can say that when the concentration of DPG is increases, the affinity of O2 for hemoglobin is decreases, because in the active tissue the higher concentration of DPG stimulate the release of O2 from oxyhemoglobin. In human red cells, the DPG is present at about the same molar concentration as hemoglobin. So you should remember, whenever the DPG concentration is very high, then the oxygen affinity of hemoglobin is decreases with the increasing concentration of this DPG. So this is all about the allosteric interaction. So at least you should know the homotropic and heterotropic allosteric interaction. It is not possible to explain in detail, but you should remember the types, that is homotropic and heterotropic. In homotropic, you should remember the cooperative effect, and for heterotrophic interaction you should remain the Bohr effect, root effect, and the effect of DPG on oxygen affinity of hemoglobin. In the next lecture we will discuss the next type of protein ligand interaction, that is the complementary interaction between protein and ligand. Thank you.